Hello and welcome back to A Level Biology Help. Today I'm going to take you through the Populations and Ecosystems section for AQA A Level Biology. Also, at the end of the video, I'll be going through a few exam questions and explain their mark schemes. And as always, there will be timestamps in the comments section so that you can skip to the different sections in the video if you do not wish to watch the whole thing. Right. So this is the basic structure of the video today. So first we are just going to go over some important definitions, then factors affecting population size, estimating population size using quadrats, transects and the Mark-Release-Recapture technique. Then we're going to go over primary succession and then the conservation of habitats briefly. And then after that we'll go on to exam questions. Right, so let's get started. So here are some important definitions. First one is community. So a community is all populations of different species in the same area at the same time. So the main difference between a community and a population is that a community is made up of different species instead of just the same species, which is what a population is. Second definition is an ecosystem. So an ecosystem is a community and the non-living components of an environment. An example of an ecosystem would be a tropical rainforest or a desert, for example. And ecosystems are dynamic and can be of different sizes. The next one is a niche, which is an organism's role within the ecosystem. Each species occupies its own niche, which is influenced by a combination of abiotic and biotic factors. Biotic factors are the impact of interactions between organisms, so for example, competition and predation. And abiotic factors are non-living factors in an ecosystem, such as weather. And the last definition is carrying capacity, which is the maximum population size an ecosystem can support. So this is a typo, it's not supposed to say and, it's just supposed to say an. So there are many factors affecting population size. One are abiotic factors. Some examples of abiotic factors are pH, soil quality, temperature, oxygen levels, carbon dioxide levels, light intensity, etc., etc. Plants and animals, so most organisms actually, are adapted to abiotic factors, which develops through the process of natural selection. If you're not familiar with natural selection, check out our video about genetic diversity and adaptation, which is AS. Species diversity and population size is larger when abiotic conditions are less extreme. This is because when abiotic conditions are less extreme, then there is a higher chance that more species will survive and reproduce, and therefore pass their alleles on to the next generation. So species diversity and population size is larger when abiotic conditions are less extreme. So biotic factors affect population size as well. An example of a biotic factor is interspecific competition. Interspecific competition is when members of different species, so a lion and I think it's some kind of leopard or a jaguar or something, or a cheetah, I'm not too sure, compete for the same resource that is in limited supply, for example, light, water, food, etc. So in this image, these two species are probably competing over some food or something. So interspecific is when there are different species. The better adapted individual is more likely to survive and reproduce. So if the lion here is better adapted, so gets to the food first, this means that the lion is more likely to survive and reproduce as it has access to the limited resource, as it is better adapted, so it's more likely to survive and reproduce, and therefore increase its abundance. So there is another type of competition called intraspecific competition. So as this name suggests, it's a biotic factor, and it's when members of the same species compete for resources, and also a mate, as obviously members of the same species interbreed. So intraspecific is when it is the same species, and interspecific is when it is different species. So they can compete for resources, so light, water, food, for example, but they also compete for mates. So when they are competing for mates, so a mating partner, the more well-adapted individuals will be able to perform a more impressive courtship display or may have features in better conditions, so more neater feathers, for example. So they will be more likely to attract a mate and then reproduce to produce fertile offspring that can pass on their alleles, which make them more advantageous in finding a mate. So this is a biotic factor that influences population size as well. Another biotic factor that influences population size is predation. So predation is basically an interaction between predators and prey. So predators consume their prey. And predation has this kind of cyclic fluctuation here. So as you can see, if I just get my pen tool out, the red line here represents the predator population and the blue line here represents prey population. So as you can see, the predator population here increases. This is because the prey population is increasing as well. So predator population increases as prey population increases because there are more food resources for the predator. So the increasing prey population leads to more food resources for the predator. So therefore increasing the survival of the predator. So their population increases. However, here the predator population is decreasing. This is because once the predator population has increased, most of the food resources from the prey has decreased. So predator population decreases as there are less food resources. But as you can see here, the prey population increases again before the predator population. So here, just slightly earlier increase than the predator. This is because the predator population decreases as they're less likely to be predated on to survive. So obviously the decrease in predator population due to there being less resources means that the prey population increases again because they are less likely to be eaten. And over time this follows the same fluctuation, so it fluctuates all the time. So you need to find a way to measure population size, but this is mainly through estimation, as it will be too time consuming and difficult to measure the exact population size. So you must estimate it instead. So there are different ways. So using quadrats, belt transects, and also the Mark-Release-Recapture technique. So first I'm going to talk about quadrats. So quadrats are these kind of sometimes metal or plastic squares with squares within them, which you might have used in a required practical. So quadrats are used to sample slow or non-motile organisms. This is because they can't move out of the area in the quadrat. So slow or non-motile organisms could be daisies or different types of plant species, for example. So the first step in using a quadrat is that you must use random sampling. So in quadrats you need to place two tape measures at right angles so you can create a gridded area to take samples from. So you need to define your area where you're taking your samples from. The next step is that you need to use a random number generator to generate random coordinates to take samples from. This avoids bias in your sampling. The first step is that you place a quadrat on the coordinate. So mainly in quadrat sampling you place the bottom left hand corner on the coordinate and either count the abundance of the species, so the number of species in the whole quadrat, or calculate percentage cover, so how much of the quadrat is covered. And repeat this at least 30 times to get a representative sample of the whole population. As for example, there might be a lot of daisies in one area, but not a lot at all in another. However, sometimes you might have to use a belt transect along with a quadrat. So the belt transect can be used instead of random sampling. So here we have a man measuring estimating population size using a belt transect, which is a long piece of tape that you place along population. So a tape measure, or the transect, is placed through a non-uniform ecosystem, so for example a rocky beach. So here, belt transects are crucial as it will be difficult to place a quadrat here. The quadrat is placed at intervals along the transect to measure the impact of changes in the environment, as obviously when an ecosystem is non-uniform, the abiotic factors will probably be different. So the purpose of using a belt transect is basically to measure the impact of changes in the environment on population sizes of different species. So there are, I think, 3 different ways in estimating population size when using quadrats, whether that be just quadrats or using a quadrat and transect. The first method is frequency, which is basically how many squares a species is present in. Then we have density, which is when you just count every individual of the species. So density is basically the same as abundance. And then percentage cover, which is when you estimate the percentage of the whole quadrat. This isn't meant to have an E here, sorry about that. That is covered by the species. Percentage cover will be made easier if you have a quadrat that has 100 squares. So the next technique in estimating population size is using Mark-Release-Recapture technique, which is used for motile organisms, so organisms that move quickly, so insects. The reason that we use this for motile organisms is that if you have a quadrat for motile organisms, the organism may move out of the quadrat whilst you're sampling it, so therefore leading to inaccurate results. So the first step in Mark-Release-Recapture is that animals are trapped and they are marked with a substance that must be non-toxic, so can't kill them, weather resistant, so can't be washed off by rain or wind, and the substance that is used to mark them must not impact predation or reproduction. So the marking substance can't make them more vulnerable to predation or increase their chances of reproduction, as this will produce fluctuations in the population size, which we do not want in our experiments. The second step is that the marked animals are released and left to distribute themselves in the population. Then they are recaptured a few days later using the same trap, and a second sample of the population size is taken, and this is repeated at least 10 times to get a representative and reliable result. And we can estimate the population size using Mark-Release-Recapture by using this equation. So the size of the first sample, so when the animals are first trapped and marked, multiplied by the size of the second sample, so when when the organisms are recaptured a few days later, divided by the number of recaptured organisms that have been marked. So this is how you can estimate population size using Mark-Release-Recapture. However, the Mark-Release-Recapture technique is based on a few assumptions. So obviously the assumptions that the marking substance is non-toxic, weather resistant, and doesn't make them more vulnerable to predation or more likely to reproduce. But there are also some other assumptions. So the first one is that the technique gives the organisms enough time for organisms to distribute themselves evenly amongst the population. No migration, so they don't leave the boundaries of the investigation, and that there are no births or deaths, which will affect the population size. So now we are going to learn about primary succession. So what is succession? What do we mean by succession? Succession is a change in an ecological community over time. So when the species change over time. The first step in primary succession is a pioneer species colonizes bare rock or sand, so an area where there is a very, very low species diversity. So a pioneer species is the first species to colonize a piece of land. The pioneer species are adapted to survive harsh abiotic conditions. And these pioneer species can change abiotic factors to become less harsh through their death and decomposition, which releases nutrients into soil. So the pioneer species become decomposed, which releases nutrients into soil, so decreasing the harshness of abiotic conditions. The next step is that moss and smaller plants can now survive due to the nutrients in the soil and the less harsh abiotic conditions. And when these moss and smaller plants die and decompose, they further increase the depth and nutrient density of the soil as they die. And this process continues and continues, so larger plants can survive as the abiotic conditions are becoming less and less harsh. So this further changes the environment. And the new species that are coming in may change the environment in a way that prevents this previous species surviving as they become outcompeted. So larger plants may block the sunlight from the smaller plants, for example, so the smaller plants die. So they become outcompeted so they can no longer survive. These changes result in a less hostile environment. The term less hostile here is key. And increased biodiversity. And primary succession results in what we call a climax community, which is when the community is made up of mainly one species, which are mainly trees. So here we have an image which represents a cycle of primary succession. So here we have step 1, so rocks, which have a primary or pioneer species, which make the environment less harsh, or the abiotic conditions less harsh, so larger plants can grow as the pioneer species dies and decomposes. This process happens again with the second species, so larger plants can grow, which can then outcompete the previous species. So larger and larger plants can grow, which results in a climax community, so mainly made up of trees. So habitats, so areas in which organisms occupy themselves, need to be conserved. So to conserve habitats, the process of succession is often managed. The example would be the grazing of cows in a field. So the cows eat the grass so to prevent the nutrients being released into the soil, so preventing succession occurring. So by doing this, so by managing succession, a climax community is prevented. So there is a wider variety of habitats and therefore a higher species diversity. However, this creates a conflict as human needs and conservation clash. So conservation is needed to create a high species diversity. However, human needs increase the need for things like food, so in monocultures. So to balance this, an example would be to coppice trees. So coppicing trees is when trees are cut back to their stumps. This process allows timber production for furniture, so providing human need, but this still allows survival of the tree. So here's an image. So this is a tree to be coppiced. So the tree is then coppiced, so cut back to their base in the winter, so to provide wood for furniture needs. However, this means that the shoots can also grow rapidly, so therefore conserving species diversity. Right, so that is it for the content, and now I'm going to get on to some exam questions. So let's look at the first question. So ecologists use a method called proportional sampling, so it's not random sampling here, to estimate the population size of an animal species. This method is based on assumptions. Two of the assumptions are given below. So the first assumption is they know the size of the area A, so the A is the size of the area, where the animal population lives. And the second assumption is that the animals are uniformly distributed in this area. So to carry out this method, the ecologists chose a region of known size R inside area A. So the size of the region is known, which is crucial in the investigation. Then they counted the number of animals in region R. They called this number S. So S is the number of animals in region R. Then they assumed that the number S would be in proportion to the size of the total population P in area A. Proportional sampling can be used to estimate the population size of a species that is uniformly distributed. So the question asks us here, what is a species? So this is a 1 mark question, so it's basically asking you for a simple definition of species. If you aren't familiar with what a species is, please check out my video about species and taxonomy. So a species is organisms that can interbreed and produce fertile offspring. So let's look at the mark scheme. So it says organisms that, so you don't need to actually put this phrase as it is in brackets. So organisms that can breed together, interbreed, and produce fertile offspring. So as the word "and" is in bold here, you need to put both of these points to get the mark. So you need both aspects. And it rejects inbreed. So if you put anything referring to inbreeding or viable offspring instead of fertile offspring, you do not get a mark. So let's move on to the next question. What is meant by uniformly distributed? This is a pretty simple question. When something is uniform, it basically means that it is all the same or it is even. So I've written that the organisms are equally spread. So let's look at the mark scheme. The mark scheme says you can either put the same number of organisms in each region or that the organisms are equally spread. We wrote that the organisms are equally spread, so we would get the mark for this question. Also, it allows other ways of expressing region or equally spread, for example, not clumped together or the same number per unit area. So you can refer to other ways of expressing these phrases to get the mark. The next question, I've just copied the information from the first part of the question here, says use the letters A, R and S to write an equation showing how proportional sampling is used to estimate the total size of a population P. Show your working. So you need to use A, R and S to create some sort of equation to calculate P. So P is the total size of the population. So remember that the size of the area is A. They have chosen a region to sample called R, and then the number of animals in the region are called S. So this is the equation that I've come up with, A times S divided by R. I've written this because if you use Mark-Release-Recapture, you need to multiply your samples. So you need to multiply the area, so the size of your area, by the number of animals in the area by the region that you are sampling to get a total size of the population. So let's look at the mark scheme. So the mark scheme here says P equals AS, so A times S divided by R. So we would get that 2 marks for the correct answer. Also here it says 1 mark for having A on top of the equation, and it says here recognises that total population is related to total area. So as A is total area and you're calculating total population, A needs to go on the top. Also it says here, note P equals A times S over R or P equals A divided by R times S are also correct. So we actually wrote A times S over R, so we would get both marks still. Also it says allow 1 mark for S over P equals R over A, as these equations are equal, however it is asking you to use A, S and R in the same equation, so you can only get 1 mark for this. So here is the next question. Population size can be estimated using proportional sampling or Mark-Release-Recapture. How do the assumptions made in proportional sampling differ from those made in Mark-Release-Recapture? So if we go back, the assumptions in proportional sampling is that the animals are uniformly distributed and that they know the size of the area. However, in Mark-Release-Recapture, there is no assumption that organisms are uniformly distributed, and the size of the total area isn't required, as I didn't mention these when I was talking about assumptions of Mark-Release-Recapture. So there is no assumption that the organisms are uniformly distributed. I just mentioned that they are just distributed. And the size of the total area isn't required, as you are just calculating the population size when you use Mark-Release-Recapture. So let's look at the mark scheme. So in Mark-Release-Recapture, or you can write the converse, so the opposite to these if you're referring to proportional sampling. The first marking point, no assumption that organisms are uniformly distributed, so we would get a mark. And the second marking point, the size of the total area / the size of the sampled region is not required, which we put, so we would get all 2 marks for the question. Here it says marking point 1 or marking point 2 do not have to start with the same technique. And also, in this case, allow difference by implication, i.e. do not penalise if the 2 techniques are not compared. So you don't need to compare the 2 terms to get marks. Also, as it says, the marking points do not have to start with the same technique. You can write 1 point referring to Mark-Release-Recapture, and you can write the next point in the converse, so referring to proportional sampling to get the marks as well. Right, so let's move on to the final question in the video. So it says, give one assumption about the animals caught that is made in both methods. So I've put that the animals are all from the same population. I don't think I mentioned this in the Mark-Release-Recapture section of the video, but an important assumption in sampling is that the animals are all from the same population. You don't need to go into any more detail, as this is only a 1-mark question. So let's look at the mark scheme. So the animals all are from / all part of the same population, so we would get the mark. Right, so that is it for this video. Thank you very much for watching. If you have any questions, big or small, please leave them in the comments. I'll be more than happy to answer them, and I'll see you in my next video.